Thank you, Sarah, and thank you for the opportunity. I'm not the principal sponsor. You people out there are, because the consortium's funds comes from farmers and it's matched with the government. But it's a great opportunity to be involved, and we have the opportunity to promote this great work that we've done in genetics. The consortium's been around since 2003, and we've driven $85 million into finding ways to reduce methane and nitrous oxide in our herds and flocks. And in 2003-4, there was actually 700 dairy cows, which is the bottom picture, if you haven't worked it out, that were actually tested for methane and to see whether we could find breeding, and whether actually in those days whether there was any variation to be actually used. We did find some variation, but we had really piss poor intake measurements, and we really didn't have that good of methane measurements, and so that kind of work wallowed for a while until, and we used SF6 for the sheep and deer to sort of work out their main emissions, but it wasn't until 2007 when we moved from using SF6, the little halters around their neck, to chambers, and then found that there was some variation, and the sheep program was kind of born. And so we really got two people to thank for that, in the sense of driving a big team of researchers that have done it. John McEwen is here as well, and he started it in the sense of once we started it, there was variation, and Suzanne Rowe, who you're going to hear from shortly, has picked it up from there. But I just wanted to also take the opportunity to give you a little bit more background to the other genetics things. So not only have we gone down the path, and you're going to hear about the sheep genetics, but we looked at cloned cattle and fasciculated cows. We looked at the 700 cows. We investigated a bloat herd. We screened to get to the sheep you'll hear about today, and breeders are now using 1,300 sheep through respiratory chambers. And we've used a whole lot of reasons to get, and used the new science to get there. So it's been a real big team game. We've been lucky enough to be the ones who've been funding it and supporting it, along with the Greenhouse Centre. But I also want to acknowledge that when we found out that the variation could be found in respiratory chambers, we then focused on the central progeny test, and it was the industry that says, let's have a look at the females in the central progeny test, which in those days, 2007, I think the males were there for meat and improving meat traits. But really being able to put those through, and these flocks that are named up here, I take my hat off to you. I did actually have a cloth poster I was going to wear, but I couldn't find it this morning. So, because really the reason we're here today talking about methane selection is because you people have made those animals available into the CPT, and I acknowledge that's all a lot of breeding and a lot of passion and championing of your breeds to get to there. So that's really important that we're taking everyone with us. I'm going to introduce Suzanne now, but I just want to say that we've been rolling out the pack chamber, and we've been doing selection, and a lot of you breeders in this room have put some of your animals through it and have booked in. We have about 10,000 animals, and that's just the cross-section. You'll hear a bit more about that in a minute from Suzanne. But it's really good that it's taken a while, and it's a slow, hard game, this genetics. I'm not telling you guys anything you don't know. But really, when we look at greenhouse gases, 48% of our emissions in New Zealand come from agriculture, and 80% of that is methane. And farmers don't have too many tools. This is one of the tools that we've been able to get through and roll out. We have others under development, but this is the one that we've got right in front, and the dairy industry and the cattle industry are trying to chase it along. So without any further ado, I'd like to just hand on to and introduce a senior scientist from AgResearch, Dr. Suzanne Rowe, that has been really foremost in really driving this and picking it up. And I'm sure we're going to hear lots more about what the future might be as we move from pack chambers and direct measurements and other things into the future. So welcome, Suzanne.
Awesome. Thanks, Mark. Can you hear me?
Yeah, awesome. Dave says you can hear me. Okay, so welcome to the worst case scenario. This isn't a single gene trait. It's not even a multi-gene trait. It's definitely polygenic. So there have been a huge amount of challenges, but I'll run you through the program give you a few insights from a conference that I attended a couple of weeks ago, the Government Climate Change Conference, and what the Climate Change Commission have come up with in terms of challenges for us as an industry. Some of the scientific tools we've been driving to try and get there, and some of the things that we think are coming that we can have in our toolbox. So this is basically enteric methane. I'm going to talk about methane emissions from livestock. I'm going to give you some numbers, but as Mark has alluded to, 80% of the methane in New Zealand comes from ruminants, and it's around 35% of all GHGs actually come from grazing livestock. So it's a big deal. Is that working? Oh, yeah. So how does it show up? We've got complex starches and celluloses. They're going into the rumen. They're being digested by microbes. So it's a symbiotic relationship. The microbes are sitting happily in a nice warm gut. They're breaking down the starches. They're producing volatile fatty acids, acetate, propionate, and butyrate. So that's an energy source for the animal. There's a bit of microbial protein in there too. So that's what we're using for product. But during that fermentation, hydrogen is given off. So hydrogen just swells up the rumen, stops things happening, and there's some very clever bugs, some archaea, and all they do is combine the hydrogen with carbon. It's super clever, and they just erupt it at the front end and get rid of it as methane. So it's an ancient system, it works beautifully, but no one told ruminants that greenhouse gases was going to be an issue in the future. So this is what the emissions profile in New Zealand looks like. From enteric fermentation, lots of kilotons, 35% of our gross emissions, 73% of ag emissions. Dairy cattle are accounting for 48.4%. That means it's actually quite hard to get research money for sheep now because dairy cattle are the problem. Beef cattle, 20%, so if you sum those together, most of the research now is in cattle, but sheep still producing around 30%, and deer coming up with 1.7%. And this minus figure is basically where for a while we've planted trees, and no one's very happy about continually planting trees. We've been asked to come up with other solutions, and the climate change report has come up with budgets. So they've suggested budgets to the government for how we can become zero or carbon neutral by 2050. But they have conceded that methane as a short-term gas, as a biogenic gas, is going to be really challenging. It's not going to be as easy as for example, for the transport sector. It's not just going to be about money. So we have a sort of a flexible target for 2050. We're expected to hit 10% by 2030, reduction in methane from 2017 levels, and somewhere between 24 and 47% by 2050. And we're expected to be net zero in the long-lived gases such as carbon dioxide. So when someone talks about GWP 100, or they talk about global warming potential, which is what GWP stands for, what they're actually saying to you is if I took a kilo of methane and I emitted it in a pulse, how does that stand up to the same amount of carbon dioxide, which is going to stay in the atmosphere for 1,000 years? And at the moment, our warming factor for methane is 25. So it has 25 times the warming potential of carbon dioxide equivalent over 100 years. And we've been looking at ways of, how do we think about that? Do we assess over a short time? Do we assess over a long time? Do we account for the fact that methane actually breaks down over sort of 12 to 15 years and doesn't hang around for 1,000? Does that mean we should hit methane harder because we can make more bang for buck really early? Or does it mean that it doesn't matter as much? So these are all the questions that have been going around in the government's head and have been challenged, the Climate Change Commission have been challenged to think about. And this is what they've come back to the government and basically said. By the end of this year, they have to set their emission budgets out to 2035. They have to give us a pricing strategy. It's been really hard for us as scientists to come up with a value proposition and think about the tools that we might use when we don't know what the cost of carbon is. And it's the same for everyone. We don't know what the value proposition is for breeding for methane. So we're kind of in the dark as to how much selection pressure to put on, what should it look like in an index. And the government have been challenged with coming up with a pricing mechanism to give us some confidence to move forward. They've been asked to develop advisory services. That's so people can get out there, come up your driveways and help you to know your numbers. So there's this challenge set that everyone's going to know their numbers by 2025, but everyone's going to need help to do it. It's complicated. These calculators need to be on board, and you need the help for it. And certainly Beef and Lamb have been working hard in that space. Improve rural digital connectivity. Anyone who tried the internet here over the last couple of days will know that digital connectivity, digital tools are great, but if you can't connect to them, they're not going to work. So we really need better connectivity. And the Climate Change Commission have challenged the government to actually give you that so that you can access the online tools that you're being asked to use. To remove barriers to deployment of emerging technologies, that means if something like a fancy feed comes along that we can really use, that the barriers to use that food, the food safety barriers, are high enough that we feel safe, but that they're pragmatic and things move quickly. Support systems and infrastructure for low emissions and changes in land use. Should people want to change their land use, the support systems need to be in place. And to continue to invest in R&D to deliver technology such as vaccines and inhibitors, which are very, very expensive to develop, but could yield huge results in future decades. And I just want a nod to Hewaka Ekanoa. Some people will be sitting here saying, Hewaka what? You've heard this term bandied around, and it's basically, it's industry joining with government and setting some goals and basically setting out work streams. So as there's someone working on the tool. So there's a whole group, it's a whole bunch of people, a big steering group saying, right, what does the primary industry need? How can we approach this? What are we going to get asked to do? And how can we help farmers to achieve it? So I'd encourage anyone to go onto the Hewaka Ekanoa website and have a look and see what your people are doing for you. So back to science that I'm comfortable with. This is the program that was set up 10 years ago by John McEwen. So these are progeny test flocks, as Mark talked about. They all went through a respiration chamber, so it took several years. If you put a sheep in a respiration chamber, so we put these sheep in, we could only do 24 at a time. They went in for 48 hours, they came out for 14 days, they went in for another 48 hours, and at the end of a sort of 3 to 4 year program, we had 2 distinct flocks. We had a high methane flock and a low methane flock, with the intent that we were going to breed those flocks for several generations and see if the trait was heritable. And that's what was done. This is 9%. It's moved to 11% over the last generation. We're about 3 generations in. But to cut a long story short, we've shown really clearly in sheep that methane is heritable, has a heritability of around 0.2 to 0.25. It can be measured and it can be bred for. So we can select for it. We can do it at pasture. The results are the same at pasture. That was a really, really important result. A really important result was for us to take sheep back to the respiration chambers and feed them grass and check it did the same, because the rumen is all about food, right? Methane is 80% what you feed. So if this wasn't going to work on grass as opposed to loose end pellets, then we were already in hiding to nothing. Fortunately, it did. And we could move on to look at, we've got these respiration chamber measures and we're taking pulses of methane. We're measuring every 6 minutes. Okay, so we're measuring every 6 minutes. How many of those 6 minutes measures did we actually need to create a breeding value for methane? And what we found was actually not too many. So 12 or 8 to 12 would pretty much do it. And we found that actually if we took a portable chamber, took a really simple meter and measured sheep for an hour, that actually the breeding value that we got was just as accurate when it came to ranking animals as spending $1,000 trucking an animal to Palmerston North and putting it through a respiration chamber. Again, really big step forward. Just having short-term measures and ways of using technology on farm. So we put this into the Woodlands farm in Invercargill, and that meant every sheep on farm could go through one of these pack chambers. So we had a methane value for everything, and everything that was measured for anything else, be it meat quality, be it reproduction, be it feed intake. We looked at the alimentary tract of the animals, we CT scanned them, so we had methane, we used high density genotyping, we actually sequenced some of these animals, and we built up a really, really clear picture of what an animal looks like that is low methane and what the genetic correlations are between the other production traits. And this is the sort of conclusion of the main science program when it comes to the selection lines, was that a low methane animal gave around 11% less methane per kilo of feed eaten. So that's also really important. We don't want to just breed smaller animals. Obviously, smaller animals will give us less methane. We want them to give less methane per kilo of feed eaten, per kilo of dry matter. They had 20% smaller rumens, which was a surprise. That was a big surprise to us. It shouldn't have been, because obviously, you know, this animal is in some way controlling those bugs, so there must be something going on physiologically. But we did see the same surface area, so that was really interesting. So the papillae are denser, they're longer, so we've got the same surface area in the rumen, and we didn't see any differences in passage rate, but we did see a smaller rumen when we looked at the rumen volume. When we put through the feed intake facility, they tended to eat slightly more, but they ate little and often. Now, eating slightly more means they were less feed efficient than the high line, but when it came to actual profitability, they put on more muscle than fat. So that was a bit of a complicated story, and we're still sort of trying to unpick that in terms of dollars and cents. They had a different microbial fermentation, or I guess that should have been obvious. If they're fermenting feed differently, then they're going to have different microbes in there, and those microbes are heritable. They get passed on from one generation to the other. They had a different energy profile, so there's more propionate coming out. That changes the sort of the profile of the meat and the milk. I'll talk a little bit more about that later. So we saw more lean growth, we saw less fat, less acetate, we saw more wool, again, bit of a surprise, but there must be some sort of limiting factor in those microbial fermentations that meant that something was being provided to the animal not being provided before. But in the main, they were healthy and profitable. And when we stack them up on the maternal worth index, this green line here is the low methane line. They look good. This is so the blue line's the average, the red line is the high methane line, and this is flock 2638, so the Woodlands progeny test flock. So you can see that the low line's tracking really well, mostly because they've got a little bit more growth, they're a little bit better in terms of parasite resistance, less dags, a bit more wool. So all we were interested in is that we were doing no harm. In actual fact, it looks as though there's a benefit to being a low methane animal, but we're a bit cautious about that because, you know, we've got a pretty low number of founders. But we were confident we could roll it out. We could get out there in industry and say, hey, it's time. It's time to start measuring, you guys, it's time to start measuring your animals. And we got this big trailer manufactured, which was really exciting. You'll see it on the road. It's enormous. It's awful to tow, but it's great. It goes up people's driveways. We can measure 84 sheep a day, and combined with genotyping, it's really given us scope. It's really given us scope to actually get out there, you know, and measure people's animals. And everyone in the world wants one. So we flogged them all over the place. We flogged pack trailers to Norway. They've stuck them in a truck, and they've got a coffee machine and a heater, which we're all jealous about. Uruguay's still a little bit behind. They're still using the old-fashioned method, but still using a pack trailer. They're in Australia. We sold one to Ireland. We had an email from France a couple of days ago. Scotland have bought one. So everyone wants a pack trailer, which is really exciting. And I'm actually just going to see if I can let that play. I just wanted to show you guys, for anyone who's never seen one, this is what it looks like. This is the infamous Janine, who's here somewhere. She could probably stand up. This is Barry Van Vliet. He runs the trailer. And it's actually really quite straightforward. It's as simple as getting a few sheep, allocating them really carefully, understanding how they've been fed before they go in, but then just popping them into these, and they go in for 1 hour, and that's it. We get a BV with a little fancy statistical jiggery pokery to get the moles off that machine. So there you go, Janine. Famous. Okay, so how many measures do we need? We figured out for a decent accuracy, given the heritability of the trait, what we knew about the trait, we needed 10,000 measures. So at the time, that seemed unsurmountable. But we got there, and actually we got there pretty quickly. And I just want to talk a little bit about how we got there. So back in 2017, we had large numbers being measured in 3 flocks. These are the research flocks. So we've got the blue is flock 3633. So those are the methane selection lines. Obviously had to be measured on a regular basis. We had the Woodlands progeny test flock. So we had packed boxes in the woolshed at Woodlands. It was easy to do. And we had flock 4640, which beef and lamb central progeny test. They were going through feed intake. So we had those being measured. And then same for 2018. But then we got this beautiful pack trailer built up, and in no small part, we had Barry Van Vliet. Barry is a technician for AgResearch who went away and rolled into my office one day and said, "Do you have any work?" And it was just the most fortuitous moment, because it was just at the time when we were thinking, how the hell are we going to man this big trailer? And he has been amazing, you know, working with the calibration of the machine and the fact that if you get a damp filter, you can't measure anything. And he's just been incredible. There's been so many sort of, you know, teething issues with getting something like that on the road that he's in no small part made this a success. So in 2019, we didn't have any funding for the trailer, but we had some really, really pioneer farmers step up and say, "I'll do it, I'll give it a go." And they paid for 2 measures. So they financed it, they had the trailer up their driveway, and I can't thank them enough, because they're the guys that help with all the teething problems. They're the guys that were on the phone. We worked out the breeding values ourselves. We sort of went back to them and said, well, this is what it looks like. And this really was the heart of the matter. Then Mark Aspin got involved with the PGGRC and said, hey, I can help you out. If these guys are prepared to pay for 1 measure, I'll pay for the second one. And that moved into 2020, where we had a lot more people join our happy crew. And then at the end of 2020, we sat down, looked at the data and said, do we really need to drive back to these farms? Because the logistics of getting the trailer on farm and then 2 weeks later needing to be back at the same farm when you've moved up and down the islands was just incredible. So we sat down and looked at it and said, okay, if we move from measuring 10 progeny a sire to 15 progeny a sire, we only need to go once. The accuracy is roughly the same. So that was a big move. Yeah, that was a big step change. And we introduced the single measure, and that really helped for a few more people to come on board. And we're currently in the situation, of course, where we need another trailer. It's really exciting to be in that position where we're moving up and down the country. We went to Northland and put some goats in it. The goats worked really well. So if anybody's into goats, actually goats was quite successful. And basically 42% of those 10,000 measures have been done this year. And this is just a breakdown as Mark showed, just showing, so that the full bar is basically the representative of the breed in New Zealand, and the blue is how many of them have been done already. So Perrindale, there's a bit to go. Coopworth, I'm not completely convinced about that, because the Woodlands flock is 50% Coopworth, so they're down here as a composite. So I think Coopworth are probably better represented than that. I'm not sure what other is actually. I'll need to find out, and we've not done anything in Merino to date. But it's looking really good. The program's out there, and Beef and Lamb stepped up, programmed all our models into Improve, and they're producing weekly breeding values. So anybody who joins up basically gets a BV and can rank their size for methane, so that you've got a tool, you've got something. And we've got a table here which basically shows the range in methane values. I will say we haven't put methane yield into the breeding values. So we've been selecting our selection lines on methane yield, and that's methane per unit of feed eaten. What we're delivering at the moment as a breeding value is just methane. So it needs to go into the index. It has to be set against the other traits. And the reason that we did that is because we didn't want double counting in the index. So we set it up right from the start, because we know it's going into the index, and we don't want to give a ratio trait that then has to be picked apart and put in so it doesn't double count. So currently you are only getting methane emissions, total methane emissions. So if you're using it, and we've got a workshop tomorrow, we can talk a bit more about it. You just have to be a little bit cautious that you're not getting a yield trait at the moment. You're getting the absolute emissions that your animals are giving. So smaller animals will have a low breeding value. And just going back to Flock 2638 and where we are, I think about 2 or 3 years ago, I stood up and said, right, well, I'm going to do it. I'm going to put methane into my selection index for the Woodlands progeny test flock. It felt terrifying. You know, I'd been handed this progeny test flock after John had successfully run it for decades, and it was looking great. And now I was going to put methane into the selection index, and God knows what was going to happen. But we gave it a go, and we looked at what would happen to the index, and we looked at if we chose the 22 best sons, so we got 22 sire lines ranked by maternal index. If we just chose on maternal index, we sort of anticipated we'd get an increase of about 1.7% in methane for the next generation. If we chose on maternal index plus methane, then we dropped about $1 on index, so we lost a bit of productivity, but we actually reduced methane by 3.7%. And that's really manifested. So that was at this point, and you can really see it's actually well over 5% drop in methane in that flock. So we've put our money where our mouths were, and we haven't seen a big drop off in maternal earth. Things are still going up. Methane's going down. We valued it at $100 a ton. We are seeing a slight drop off in lamb growth. So we're keeping an eye on that, and in adult size. So we're out in front. You know, we'll keep that test plot running as long as we can, and keep an eye on things, and keep reporting as to what we see. John and I ran some numbers for the Climate Change Commission. We looked at it and said, okay, so we've looked at it, we can see it works in industry. How about government steps up and thinks about what it would cost to genotype and phenotype the top tier in the breeding industry? How about we take the top 5,000 sires, make sure they're genotyped. Genotyping is getting cheaper and cheaper, and we measure them. What's the rate of return on $50, $100, or $200 for a ton of CO2? And what happens if we use global warming potential 100, or we use the split gas approach with WE? And the answer was they always made a profit. If carbon is going to be valued at over $50 a ton, then we can quite comfortably give reasons why the government would invest in the sheep flock. They're going to see a return on their investment. So we submitted that to the Climate Change Commission, and we're in talks with MPI at the moment. And this is basically what that looks like. Now, I want you to look at this number. When we did all our sort of reckoning, we said, when will adoption start? Adoption's going to be low. It's going to be about 0.3. We probably won't see full adoption until about 2028. So we're only seeing a 0.23% drop in methane early on, and we were assuming that we would see about a 0.5% drop in methane per year over the whole industry going forward. Now, because of the adoption rate, that means that at 2040 we'd still only be at 7.5%, which is enormous. It's incredible. Let's not take away from that figure, but it is only 7.5%. It's not 10%. It's not 20%. It's not 50%. It's 7.5%. It's conservative, and accounting. I think it could be higher than that, but it is there. We have a tool. We have a tool in place. It's the first cab off the rank. We know it works, and as far as we can see, it's not doing any harm. I spoke to Peter Jansen. He's the lead scientist in the program, funded by the PGGRC and the NZAGRC, and said, what else is out there? Because the government is saying we need to hit 24 to 47%, and they're saying the first 10% we reckon you can do with what you've got, but we know that other technologies are going to need to come along. So what are we looking at? And the other technologies look like this. So we've got brassicas out there and crop feeding. We know it reduces methane. We don't really know what level of feeding is required, and we don't really know how it's going to fit in the system. But that's available to us now. There's 3-NOP, I'm sure you've heard about it, and there's seaweed containing bromoform. These are feed sources that have been shown across the world to work in lowering methane in ruminants, generally being mixed into TMR diets. Not very easy to see how we're going to feed them on a pasture-based system, but if they work, we'll come up with a way. And that's what's happening at the moment. So there's development required for New Zealand systems, but that development is ongoing, and it's going ahead. The PGGRC has a really strong inhibitor program. They've had it for many years, and they're looking at developing products for pastoral grazing systems. So, a New Zealand-based solution, and there's the vaccine program. The vaccine program is really exciting. It's been successful, but it's incredibly slow. It's not like the COVID vaccine where they knew what their target was. We didn't know what our target was, and we didn't have lots of money behind us either. So this is slow, but hopeful. Could be huge response. So there are technologies out there coming, but we don't know when they're going to hit, and we don't know how effective they'll be. So we can't just sit on our hands and just hope for a vaccine one day. And finally, where are we going? Back to the sort of science program. We've been really interested in the actual microbes themselves. So we've been taking a rumen sample from everything that we can, and that's simply a tube down the throat of an animal. We use a syringe, we pull out 30 mils of the rumen fluid, and we sequence it, and we look at the composition. And we've been predicting lots of traits just based on the rumen profile alone. It's actually pretty cheap and easy to do. There's not much genome in a microbe. We sequence about 1 to 2% of the genomes, and we can predict genetic merit, and we can combine them. We can use microbes and genotyping, and it's actually working really well in increasing accuracy for things like feed efficiency and methane. So the blue bar is when we use the GRM, so that's just a straight breeding value. The orange is when I use the microbiome, so it's actually better for feed efficiency than genotyping, but you get the most bang for your buck when you combine them. So there's a few really interesting points there, and it may be that we can combine it. It may be that we don't need to drive up everybody's drive with the pack trailer. It may be that we can actually take rumen samples. And the other thing that I've been doing is we milked the selection lines. Actually, it wasn't too bad. It wasn't as hard as I thought it might be. But we milked the selection lines for 3 years, and we sent the samples off to LIC, and we did some detailed fatty analysis at AgResearch, and we looked at the microbiomes, and what we found was that the milk and the highs and the lows is really different. So there's more branched chain fatty acids, there's more conjugated linoleic acids, there's less short chain fatty acids, there's more polyunsaturates. And the variance explained by the microbes in terms of the volatile fatty acids, the acetate and the propionate, was around 30%. So that's probably driving the whole system. We see differences in lamb growth. Well, there's no wonder we see differences in lamb growth when we're seeing differences in milk. And Trish has looked at the fatty acid in meat, and we're seeing the same sort of profile. So you can flip that on its head and say, well, if we can take a milk sample and see it's different, could we use that to predict methane? Long road, difficult road, but certainly one that we're talking to the dairy industry about. As what we're really looking at now is how can we transfer some of the learnings that we've had into the dairy industry to help them on their path and to get them there a lot quicker than we've got there. So in summary, low methane can be passed on to the next generation. We've got a tool, and we haven't seen any detrimental side effects to date. We think there's potential for lowered emissions in the industry for around 0.5 to 1% a year. It's cumulative, so it's there forever. Well, not forever. We've got an accountable, measurable way to measure methanes. At this moment, I don't know how it's going to be accounted. I don't know how it's going to go into the calculators, and we thought about this. What does it mean? If I've got a methane breeding value for my RAM team, how do I fit that into my emission statement? What's it going to look like? How do I plug it into these new calculators that are coming on board? And we've been discussing in detail how this might look. And at the moment, our best guess is that we would express your BVs as a percentage. So the IPCC is counting noses. So your animals may be a bit like stocking rate. You're actually 90% of a standard flock. So we could express the number of your animals as a percentage of a whole, given that your animals are producing less methane. So your animal might be producing only 90% than their standard. And that's our best guess at the moment, but it's a guess. And next steps for us, genomic prediction, more microbial prediction, proxies in milk, and looking at these strategies and seeing if we can plug them into what we've done already. I'd love to get a vaccine into a low methane animal and see what happens. Does it go even lower? Does it make no difference in the low methane animal because we already bred the difference? We just don't know. So we're really excited about those strategies. And finally, we've got a minus 40 by 40 HFFF application on the go where we really want to push that woodlands flock. We really want to see what happens if we use every tool in our box. That's system tools, that's feeding, that's hogget lambing, that's reproduction, reproductive technology, anything that we can throw at it, digital tech, any of the new technologies we can get. Hell, if we can gene edit grass, we'll do that. Any new technology that we can have and use on a research farm, we would like to chuck at it and see if we can hit that 40%. and at the same time test and develop all these on-farm tools and calculators and see how they match up. And in the words of Hewaka Noah, really know our numbers and drive the conversation. Say, well, look, we've looked at this in a standard flock on farm. This is what the numbers look like. So this is what you can ask for us, because this is all we can achieve. And with that, I'll thank the many, many people that have come before me and funded it. Apologies if I've gone over. I didn't look, oh, the light's flashing at me.
That's great. Thanks, Suzanne. We do have a few questions that have come through on Slido, which we'll try and rip through. First up, is there much variance in methane levels between breeds?
So from what we've seen, pretty much the breed variation is the same. So we see roughly the same variation, whichever breed we look in. So we haven't seen it. We thought we might at the start. We saw a few founder effects in the selection lines at the start, and we were kind of sneaky suspicious that the Texel was giving an advantage, but that's kind of dropped off. So I think there's pretty much an equivalent amount of variation in all the breeds.
Cool, great. Can the microbes identified in the lower methane producing animals be isolated, multiplied and reinserted via an oral drench or something similar?
So possibly the sort of Yakult effect. It would be quite hard to do. I think you would have the issue with feed that you'd probably have to do it often, which is the same issue with the inhibitors. It's essentially the same things as an inhibitor. So potentially yes, but I'm not sure how effective it would be, because if there's a sort of physiological genetic basis driving those microbes, then the chances are that the animal would revert to what the status quo was.
So selecting a low methane animal will select for a small adult, high repro, more wool and less fat. Is this going to be an intensively, and I'm going to guess that's breed animal?
Selecting, let me select for a small adult, high repro, more wool, less fat. Is this going to be an intensively bred animal? The answer is I don't know for sure. We've looked at different quality feeds, and we've run them. So we shipped them all to the North Island, and we ran them on different pastures over different seasons, and we didn't actually see any difference. Nothing fell over. So at the moment, we don't have any evidence for that. I'm sure there will be a point at which a land at which they're too soft for, but at the moment we haven't seen anything that alarms us.
Are there any links between microbes in the gut and the ones in the soil?
So there's been some work on that. It's pretty difficult to characterize. Our Land and Water's doing a study where they're looking at soil, plant root, plant and cattle. So that the idea that something's being passed through, I don't know the answer to that, but it is being looked at and it is a good question.
Will you ever share your sires?
So Dan sidled up to me and managed to get a few sires out of me for his future farm this year. So I guess the answer is yes. Not from the selection lines. I think the selection lines, they're inbred. They're very specific. I don't think I'd be sending out those low methane animals, but we use an animal from the selection line into 2638 every year. So we've been taking those genetics and integrating them into flock 2638. So flock 2638 rams, I think, we could have a conversation about how to progeny test those out in industry, but not the sort of the selection lines. I don't think that would be helpful.
Per law, carbon have to be reduced per hectare per animal?
Well, that's a question for Mark.
Carbon has to be reduced. If you look at it in a plain factor, but our gross emissions, we've pledged to reduce as a nation our gross emissions. So if you look at it per hectare or per animal, that for ruminants, for agricultural ruminant systems, for methane and nitrous oxide comes from the livestock, so that's the source.
But per animal means we get to keep our animals.
Per farm, probably.
Yeah.
Per farm.
With the carbon price, sorry, you had a graph up before about carbon price at $50 a tonne, $100, $200 a tonne, and the effect. Like at the moment it's 38, and farms are going into trees, this is not like your research is awesome because it quantifies everything. But if it gets to $50 a tonne or $100 a tonne, don't worry about sheep because it's all going to be trees.
So,
Okay, so your boss has been negligent in
trees-
not putting this sort of stuff up.
trees are not the solution. The problem with trees is that you have to cut them down. So we have an issue that we planted trees to sort of kick the can down the road. Well, the can's arrived, these trees are being cut down, and we can't plant them fast enough. So, and we're not looking, or the government is not looking for trees to be planted to the beaches to solve the issue, because it's not that simple, and we couldn't plant enough trees. So I respectfully disagree that if we have a carbon price at $50 or $100, that planting trees is the only solution.
So if you can measure it with genomics, could you put a good financial case to the government to increase the uptake or through breeding by using gene editing? Because we've heard Dorian talk about the fine wool thing.
It's a great question, and if there was a single gene or even 5 genes underpinning it, I would be in their boots and all campaigning for gene editing for this trait. But we don't see anything that explains more than 1%. So it's a combination of probably hundreds of genes because it's an evolved system. So it's a really complicated system. So we can shift it using, genetics because we're looking at all the markers at once when we create our genomic breeding values. But there's nothing in there that says it would be worth the investment to try and gene edit anything of any consequence in there. So if it was a single gene or just a few genes, I'd be right with you. But at the moment, we haven't seen anything that, leads us down that path. Yeah, I know.
Cool. In the interest of time, we'll ask the few questions left on screen. Is there any variances done between levels of methane in different grass types?
So you definitely get different amounts of methane per feed type. And, we're looking at animal ranking. So at the moment, we haven't seen animals ranked differently. But yes, you can definitely feed things. And obviously, there's the GM ryegrass that's got more oil in it that's been shown to suppress methane. So I don't know what the difference is between the different sort of varieties. If you feed a high quality feed, you're going to get less methane per unit of feed. So yes, but the animals shouldn't rank any differently.
There isn't. We've tried.
Unfortunately, pasture species are terribly hard to bend. So 21.9 grams per kilogram of dry matter pretty much throughout the season. There is a bit of variation, but in terms of pasture plants, we haven't identified one that actually gives you lower methane when the animals eat it. But it's important because 85% of the methane emissions is explained by what the animals eat. In forage crops, we've got some variation, but in pasture, it's bloody hard to bend them.
Thanks, Mark. Is there going to be an efficiency proxy included in the BV or index, i.e. are you producing more land for the same methane could be considered reducing methane per kilo?
That's a question for Dan.
So probably the answer there is that that's a live question, and we don't have a definitive decision on that. Mark and the PGGRC are looking at how, some of these questions we've talked about, pricing, how these things get included into evaluations or breeding values start to get worked through. So we're at this stage with those decisions about how and where have still got to be decided. So the answer to that is watch this space really, but we'll certainly keep you up to date as to what we're thinking.